we rewind our last day's class, that is a negative regulation. By watching this video, you are able to understand how this negative regulation occurs in a E. coli cell.
Bacteria adapt to changes in their surroundings by using regulatory proteins to turn groups of genes on and off in response to various environmental signals. Francois Jacob and Jacques Monod received the Nobel Prize for their experiments, which increased our basic understanding of how the lactose metabolizing genes are regulated in E. coli. There are 3 structural protein-coding genes involved in lactose metabolism in E. coli. These 3 LAC genes are organized into the LAC operon. An operon is a cluster of genes along with an adjacent promoter and operator that control the transcription of those genes. When the structural genes in an operon are transcribed, a single mRNA is produced. This mRNA is said to be polycistronic because it carries the information for more than one type of protein. LAC-Z plus encodes beta-galactosidase, which breaks down lactose into glucose and galactose. LAC-Y plus encodes lactose permease, which transports lactose into the cell. LAC-A plus encodes transacetylase, whose function is not fully understood. The operator LAC-O plus is a short region of DNA that lies partially within the promoter and that interacts with a regulatory protein that controls the transcription of the operon. The regulatory gene LAC-I plus produces an mRNA from which is synthesized a repressor protein that can bind to the operator of the LAC operon. The general term for the product of a regulatory gene is a regulatory protein. The LAC regulatory protein is called a repressor because it keeps RNA polymerase from transcribing the structural genes. In the absence of lactose, the LAC repressor binds to the operator and keeps RNA polymerase from transcribing the LAC genes. When lactose is present, small amounts of it are converted to an isomer called allolactose, which acts as an inducer to turn on the LAC genes. The LAC genes are expressed because allolactose binds to the LAC repressor protein, changing its shape so that it cannot bind to the LAC operator. RNA polymerase can then bind to the promoter and transcribe the LAC genes.
So you have seen this video. Now this video tells that this negative regulation of LAC operon, it is carried out by a LAC repressor protein which functions there in a negative manner. So that's why it is called as a negative LAC operon control, because here the transcription is not taking place there because of the binding of this repressor protein to the operator site of the LAC operon. So that's why it is called as a negative control of LAC operon. Similarly, this operon is also controlled by a different mechanism. That mechanism we are going to see now. Now, this E. coli bacteria can go on different type of sugars. When this bacteria grown on different type of sugars along with glucose, when glucose is present with different kinds of sugars, then in such condition, this E. coli firstly utilizes the glucose. In first condition, glucose is utilized. After the utilization of glucose, then other sugars, they can be utilized. Because glucose, you know that this glucose is the prime source of energy and carbon for every living organism. Similarly, in case of E. coli, it is also a primary source of carbon and energy for the E. coli. And there will be no additional requirements for the utilization of glucose as a source of carbon and energy. While utilizing the other sugars such as lactose, galactose, arabinose, in order to utilize these sugars as a carbon and energy source, this bacteria requires production of additional enzymes along with the EMP pathway enzymes. So whenever there is a different sources of carbon and energy are there along with glucose, E. coli firstly utilizes the glucose, while the other catabolic operons, they are repressed in this condition. When glucose is available in tremendous amount, in such condition, other catabolic operons, they are not expressed. They are repressed condition. And this is why this E. coli uses glucose as a prime source of carbon and energy. It's because of a mechanism. That mechanism allows that E. coli to preferentially use the glucose as a carbon and energy source than that of other energy and carbon source such as lactose, galactose, arabinose. And that mechanism is known as a catabolite repression. And this is a mechanism of global control that decides between the different available carbon sources if more than one is present. So, E. coli, whenever there is a presence of glucose is there in the medium, it firstly utilizes the glucose, and then later on it utilizes other carbon source and energy source which are available in the surroundings. And this is possible in this bacteria because of this catabolite repression mechanism. Now, this E. coli, when various sources are available, various carbon and energy sources they are available in this bacterial environment, in such condition, firstly this E. coli utilizes the glucose and later on the other carbon and energy source. Now due to this condition, it shows diaphysic type of growth. It shows diaphysic type of growth because of the presence of two different sugars, and in that two different sugars, glucose is one of the sugar which is present which is available in that surrounding. Then because of this bacteria E. coli, it shows a diaphysic type of growth. Means firstly this bacteria utilizes the whole glucose which is available in the surrounding, and after that it undergoes a lag period. Then again after the lag period it utilizes the other sugar which is available in the surrounding, and because of that this E. coli bacteria shows diaphysic type of growth. Now this catabolite repression, it is also sometimes called as a glucose effect, because this effect was firstly observed because of the glucose. Means whenever glucose is available in the surrounding medium, at that time this bacteria firstly utilizes the glucose as a carbon and energy source, and then later on it utilizes the other source which is available there in the surrounding medium. So that's why it is also called as glucose effect. Now we have already seen this. Now this lac operon, as we know that this is also controlled by negative regulation pathway or way. In the negative regulation system, there is a production of three enzymes are there for the utilization of lactose as a carbon and energy source. But this is possible only when this lac operon, it is expressed only when there is no carbon source is available in the surrounding medium. If glucose is available, then with the help of catabolite repression, the lac operon, it is repressed. because of the catabolite repression mechanism, this lack of operon, it is repressed there. And because of that repression, there will be no production of beta galactosidase enzyme, beta galactoside permease enzyme, trans-acetylase enzyme. These enzymes they are not expressed when glucose is available in the surrounding medium. And this is also possible because of the catabolite repression. And whenever this glucose is depleted or utilized totally there, and then in such situation, the lactose is the only carbon and energy source for the E. coli. In such condition, then this E. coli bacteria starts expressing the lac operon gene and produces these three enzymes in order to bring this lactose as a source of carbon and energy and utilize it as a source of carbon and energy in order to produce that. Secondly, this lac operon, it is also controlled or regulated by a second regulatory protein. That protein, it is known as a CAP, that is catabolite gene activator protein. And this protein, catabolite gene activator protein, it is involved in catabolite repression mechanism. Means whenever glucose is available, this CAP allows utilization of glucose and repress all the other catabolite operons. Means lactose utilizing operon, it is repressed there, arabinose here repressed there. So this is possible because of this CAP protein. Now the CAP protein, it functions in a global regulatory network that allows E. coli to use glucose preferentially over all the other carbon and energy source by a mechanism. That mechanism we call it as a catabolite repression mechanism. So because of that, this catabolite repression mechanism, and this catabolite repression mechanism is possible in this E. coli bacteria, it's because of this CAP protein. So because of this protein, this bacteria E. coli, it uses the glucose as a energy and carbon source. How this works? Now this catabolite repression protein, it is also known as activator protein, and it is involved in the positive control of lac operon. Why it is called as a positive control? Because after the binding of this protein, after the binding of this catabolite activator protein, gene activator protein to the DNA, RNA polymerase is capable of easily binding to the promoter. Means this protein functions in a positive manner. Instead of repressing the lac operon, it enhances the production of lac operon gene expression. So because of that, it is also called as a positive control. Now this catabolite activator protein, it is also called as a cyclic AMP receptor protein, because this catabolite activator protein, it binds cyclic AMP, and when it binds to the cyclic AMP, then this catabolite activator protein, it is in active condition. It is in active condition. Then and then it can bind to the specific binding site which is present nearer to the promoter, and after it get bound to that specific binding site which is present nearer to the promoter, it enhances the binding of RNA polymerase enzyme. Now the CRP protein, it is a homodimer which is having molecular weight near about 22,000, and it has a binding site. Two binding sites are there, one for the DNA and other for the cyclic AMP. So this CRP protein, or catabolite gene activator protein, it has two binding sites, one for the DNA molecule and other for the cyclic AMP molecule. Now this CRP protein binds to the DNA as a dimer, and binding is mediated by a helix morphed within the proteins and DNA binding domain. Just a second. Now the cyclic AMP, it plays a very important role in order to regulate this lac operon, because the concentration of the cyclic AMP decides whether the CRP protein get bound to the specific location nearer to the promoter, that is we call activator binding site. And this activator binding site, in this activator binding site, the CRP protein, it is only get bound when it is earlierly bound to the cyclic AMP. If cyclic AMP is not there, the CRP protein, it is not capable of binding to that activator binding site present on the DNA molecule. If it is not bound there, then RNA polymerase enzyme is not able to bound to the promoter and initiate the transcription process. So in order to initiate the transcription process, CRP must be bound to that activator binding site present nearer to the promoter of that lac operon. Now the CRP protein, it is a allosteric protein. It has binding site to the DNA, but only when the CRP get bound to the DNA when it is earlierly bound to the small molecule that we call cyclic adenosine monophosphate. So when it get bound to this cyclic adenosine monophosphate, this CRP protein is capable of binding to the activator binding site which is present near to the promoter. And this concentration of the cyclic AMP, it decides the binding of CRP to the DNA molecule at that activator binding site. Now the cyclic AMP, it is synthesized from ATP molecule and by an enzyme. That enzyme is known as adenylate cyclase enzyme. This is the enzyme which forms the cyclic AMP molecule by using ATP as a source. If glucose is there in the surrounding of that bacteria, in this condition, the synthesis of cyclic AMP, it is inhibited. Means this adenylate cyclase enzyme, it is not capable of forming the cyclic AMP when glucose is available in the surrounding environment of the bacteria. Means preferable energy source is available. In such condition, the cyclic AMP, it is not formed by this enzyme from the ATP molecule. And it also, presence of glucose, it also enhance the transport of cyclic AMP from cell cytoplasm to the outside of the cell. Okay, so the presence of glucose affects the production of cyclic AMP, and it also transports the cyclic AMP out from the cell. So presence of this glucose molecule is very important in order to regulate this lac operon by positive control mechanism. And the cyclic AMP, it is formed from ATP molecule. And as you know that in glycolysis, TCA cycle and electron transport pathway, these are the three steps through which large amount of ATP molecules are generated. And this is the energy generating mechanism in a bacterial cell as well as in other living organisms. So presence of glucose carry out these three pathways in order to generate tremendous amount of ATP. If ATP is there, and you know that this ATP is the currency for energy for the all living cells. Now when glucose enters the cell, the concentration of cyclic AMP is lower, and this protein, CRP protein, cannot bind to the DNA molecule, and that results in a inhibition of binding of this RNA polymerase enzyme to the promoter, and which results in a catabolite reparation. If no glucose is there, if glucose is absent in the surrounding medium, then the concentration of the cyclic AMP, it is very high. When glucose is absent in the surrounding medium, in such condition, in E. coli cell, there is a high concentration of cyclic AMP is there. And this cyclic AMP then binds to the CRP protein. And this two complex, CRP protein plus cyclic AMP, they bind to the activator binding site, which is present nearer to the LAC promoter. And because of the binding of these two molecules at the activator binding site, it enhances the binding of RNA polymerase enzyme to the promoter of this LAC operon, and this LAC operon then expressed and transcription process enhanced 50 times more. Okay, 50 times more response is there, and the transcription process starts, but there is condition is there. Glucose is absent, but the lactose sugar must be present in the surrounding medium. Then and then, this LAC operon is expressed. If lactose sugar is also not there, glucose is not there, any other carbon and energy source then utilized. If glucose is absent and lactose is present, then the LAC operon is expressed, and these three enzymes, they have been formed: beta-galactosidase, permease enzyme, transacetylase enzyme. These enzymes, they are produced only when glucose is absent and lactose is present in the surrounding medium. Now these two, as you know that CRP and CAMP complex, it regulates this LAC operon in a positive manner, while this LAC repressor protein, it regulates this LAC operon in a negative manner, but in response to the lactose. If lactose is available and glucose is not there, then LAC repressor is not able to bind to the operator. So transcription process begins, and the transcription of all these three genes takes place to form a polycystonic mRNA, and that from that polycystonic mRNA, these three enzymes, they have been formed. And this CRP-CAMP complex, it regulates this LAC operon in a positive manner, and it is in response to the level of glucose. If glucose level is low, then the concentration of cyclic AMP get enhanced, and that enhanced concentration allows this cyclic AMP get bound to the CRP protein, and the CRP protein that binds to the specific point which is present at the DNA, on the DNA molecule nearer to the promoter, that is activator binding site. And these two, LAC repressor protein and CRP-CA cyclic AMP, they help each other in very short manner, in very little manner. Here there is an effect of each other, these two having an effect on each other, very little effect is that they are having interconnection between these two. Now once this CRP and cyclic AMP bound to the activator binding site, then it begins the natural transcription process, that is there is a binding of RNA polymerase hollow enzyme to the promoter, and this binding of the CRP protein to the DNA molecule, it is facilitated by the alpha subunit of RNA polymerase hollow enzyme. This alpha subunit of RNA polymerase hollow enzyme, they interact with CRP protein And with this interaction, this enhances the binding of RNA polymerase holoenzyme to the promoter, and this promoter then get open on the DNA molecule, and there is a beginning of the transcription process. 3 genes, they have been transcribed into a polycystonic mRNA, and from that polycystonic mRNA, it is translated to form 3 different genes: beta-galactosidase, permease, and transacetylase. So, in order to induce this lac operon, there is requirement of lactose and lower concentration of glucose and increased binding of cyclic AMP to the CRP. So these are the 3 requirements where to this lac operon, it is controlled at the transcriptional level. Okay, not at the translational level. It is controlled at the transcription level. Binding of this RNA polymerase enzyme to the promoter, it is enhanced and decreased. So you can observe this. This is the figure. This is a CRP protein which binds to the cyclic AMP, and this is the site. This is the site where this CRP protein binds and enhances. But there is also a condition is that lactose should be present along with glucose. When the concentration of glucose falls, then there is an increase in cyclic AMP. The cyclic AMP binds to the CRP protein, and this complex then get bound to this activator binding site. If this operator, you know that this lac repressor protein, it get bound to the operator when glucose is available. When glucose is available in the medium, in such condition, it is already bound to this operator. When glucose level falls, these two combine, they get bound to this activator binding site, and some molecules of lactose permease and beta-galactosidase are there inside the bacterial cell. They take the outside lactose inside the cell, and they convert this lactose into allolactose. This allolactose, it is the inducer which bounds to this lac repressor protein which is already bound there. So because of that, binding of this inducer molecule to the repressor, this repressor get dissociated from the operator. Then operator is free as well as promoter is free. Then comes the RNA polymerase enzyme which bounds to the promoter side, and it begins the gene expression of this lac operon or structural genes of the lac operon. Transcription takes place, mRNA is formed which is polycystonic. From that, these 3 enzymes, they have been produced in tremendous amount in order to utilize lactose as a carbon and energy source when there is no other energy sources available in the surrounding of that E. coli. You can also observe this in this video.
Many bacteria grow on glucose before they utilize other compounds such as lactose as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by 2 regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site. RNA polymerase can now bind to the promoter and carry out transcription.
So this is all about the control of lac operon in a positive manner. So these two regulatory mechanisms, they function together in order to finally manage the requirement of the energy and carbon source to that E. coli cell. And that's why this E. coli bacteria, now there are various operons are there inside the E. coli cell that allows this E. coli cell to survive in different environmental conditions, and in which condition this bacteria is present. According to that, these